your screen also recording will come, right? Yeah, then only in this chat box you can please put your attendance. Just present, sir, or any other things. It will record your attendance here in this chat box. Am I clear? In this chat box you can write your registration number or whatever may be. I can use this chat box data for your attendance once the class is over. Can you start writing? Do you understand? Yes. Yeah. All of you may please finish this attendance part quickly. Now I can see few faces are on, but some people are still switched off your camera. So let me introduce myself first. Probably in the introductory day, you know me by name. I am Dr. Biswanath Mahanti from West Bengal. I have done my master's in Jadavpur University, Calcutta, in biotechnology. I have done my PhD from IIT Guwahati in 2009. Then I have worked in Thapar University for 1 year. Then I visited South Korea, University of Ulsan for 1 year as a research professor. Then another 4 year as an international professor there at Incheon, Ina University, Incheon. So I was there for 6 years in South Korea. Then in 2016, I joined here at Korean Institute of Technology and Sciences now as assistant professor. Now I am associate professor of this department. I am having one scholar who is undergoing research now. We had a couple of master students. We also have to promote the research activity, what we do in the lab. And I work actually in multidisciplinary and I'll say rather interdisciplinary domain. When I was doing my PhD, that time I was working on bioremediation of contaminants, particularly pyrene. Then when I was doing for postdoctoral position, I was working on anaerobic digestion of industrial sludges, anaerobic digester, biogas production. In positions at Ina University in Incheon, I worked on carbon capture and sequestration, methanol production from biogas, mostly environmental and allied area, of course. We can have soil contaminants, soil remediation, acid soil neutralization. These were the domain I worked. Apart from that, I also worked on some chemometric techniques like principal component analysis. This is I'm very much inclined to do a modeling of bioprocess, environmental process, as Muthukumar Sara was telling some that day as introductory statement. I work on modeling of bioprocess systems. In fact, I take one course for master's program here, MTech program. I am having one designed course that is called bioprocess modeling and simulation. So my research work here also do on the same line for bioprocess, environmental bioprocess, biopolymer productions in the lab. You can visit my profile for knowing the research area and maybe for better clarity on this. So this is from my side. I want to know about you also. Can you give very brief introduction about your background and where are you from before I formally start this academic portions? So Krishna Kumar,
Yes, sir. Good afternoon, sir.
where are you now? In marketplace somewhere?
No, sir. I'm in near to my home only, sir.
But not in home, right? Yeah.
Yes, sir.
Yeah. You tell me.
Sir, myself Krishna Kumar.
Mm-hmm.
I'm from Viluppuram district,
Mm-hmm.
and I studied UG BSc Biotechnology in Guru Nanak College, Chennai.
Mm-hmm. You
Yes, sir.
in BSc biotechnology. Okay.
Yes, sir.
Nidha.
Is myself, I'm Nida Razmin, and coming from Thrissur district, which is in
Mm.
Kerala. I completed my graduation in biotechnology from Kerala here itself. Thank you, sir.
Biotechnology. Okay. Okay.
Yes.
Kiran, you're from which place?
Good morning, good afternoon, sir. I'm Kiran. I'm from Bhilai, Chhattisgarh. I have completed my BSc Biotechnology from itself.
From where?
Bhilai, Chhattisgarh side.
Yes,
Sir.
yes. Which institute, university?
Rungta Institute of Science and Technology.
Likita, right? Likit, sorry.
Yes, sir, it's Likit. Hi, sir.
Hi. What's your first name? Badugu, right? Maybe I'll be pronouncing wrong, so excuse me if I'm
Likit
pronouncing it
is
wrong.
fine, sir. Likit is
Fine.
fine.
Likit, tell me.
Yes, sir. My name is Likit, sir. I'm from Tenali, Andhra Pradesh. So I've completed my bachelor's in BSc Biotechnology.
BSc Biotechnology.
Yes, sir.
Avinya, right? Yeah, can you hear me, right, Avinya?
Yes, sir.
There is so much background noise, right?
I can't hear you, sir.
You cannot hear me,
No, sir.
but we can hear you. You may continue about your introduction. I know there is a noise there in your background, right? You may wait for some time. Then can we go for Pavitra? There is two Pavitra, right?
Yes, sir.
Yes, Pavitra, tell me.
Sir, I
Which?
am Pavitra from Kadalur. I did my UG BSc Biotechnology in Bishop Weaver College, Trichy.
UG Biotechnology. Degree is BSc Biotechnology, right?
Yes, sir.
Another one, Pavitra.
Hello, sir. This is Hulda.
Hi.
Hulda Pavitra Sagarika. I'm from Hyderabad, Telangana. I did my UG in BSc Zoology in Madras Christian College, Chennai.
BSc in Zoology.
Zoology, yes, sir.
Okay. Beula Biju, right?
Yes, sir. My name is Biola. So I'm basically from Bangalore. So I did my BSc in biotechnology, graduated in year 2018.
Who are the all other seven? I cannot see you, or maybe can you switch on your videos and just introduce yourself? Dilip is there. Dilip, Hemavatan, right?
Hello, sir. I'm Aman. I did my UG from Bhopal, MP. I'm basically from Betul, MP.
Betul MP, yes.
I did my degree BSc in BSc Chemistry honors with biotechnology as secondary subject, sir.
Okay, BSc Chemistry honors and biotechnology was your secondary. Now you are moving into masters in biotechnology, right?
Yes, sir.
Good. Who are the other one? Can you please introduce?
Sir, myself Akash.
Akash, yes, Akash.
I did my. I'm from Chennai. I did my UG, which is BSc Biotechnology in Chennai.
BSc Biotechnology in Chennai.
Yes.
Shweta is there, right?
Yes, sir. Good afternoon, sir.
Good afternoon, Shweta.
I'm Shweta from Tirunelveli. I completed my UG BSc Biotechnology in Triple V College, Virudhunagar.
BSc in Biotechnology. Okay.
Yes, sir.
Who is? We are having 17 member. Who is the next one? Anyone? I cannot see the whole list.
Sir, my name is Shakti. I'm from Chennai. I pursued my UG program in Pondicherry Acharya Arts and Science College.
Okay, this was your program was BSc Biotechnology, right?
Yes, sir.
So, who is the next one? Dilip is present. Can you please introduce? I cannot find all the participants here.
Hi, sir. I'm Dilip Kumar. I come from Pudukkottai.
From Pudukkottai, right?
Yes, sir.
And BSc Biotechnology, right? Dilip?
Yes, sir. I completed JV College Arts and Science.
So I think, it will be very difficult for me to recognize all of you because all now on online. But in another 2 weeks time or 3 weeks time, you are coming in campus. You know the information, right? First year PG. So anyway, you are lucky enough because we had one batch of students who are going to graduate completely online. They have joined in the Corona period and they will be passing out. Probably in the last semester, we will have them in the campus when they are doing projects. So for MSc and MTech program, we are also having one project semester. But for MSc programs, it is not the entirely projects. You can also have some theory, right? Theory means I mean to say some lecture classes will be there at the end same. But for masters in technology, biotechnology, they will all have a complete semester on projects. And projects are very important. And as you might be knowing, you have studied BSc in biotechnology, and finally you are coming into MSc in biotechnology. There must be some reason behind that. If you say there is what could be motivating you to join MSc biotechnology, I'm not asking why you joined here. That is a different question. Why you choose biotechnology and MSc biotechnology? There could be some important reason. First of all, maybe very simple. Some people will say, I have nothing to do, so I simply join. If some of you are in this category, you are welcome. You can have this. But that is not definitely the answer or response. You should have some life goal. If you are coming to master's program here, I believe, as it is my personal belief, I believe that the students, if I'm going for masters, I will want to see them in further, maybe in PhD program also doing some research. I would like to say, if simply completed BTech program and you are doing some job, it's all good, nice. BTech or BSc biotechnology, you are going for some job, it's good. You are coming to MSc and going for some job, it's okay. But once you complete MSc and you have good orientation on work, you are doing some lab exposure. In BSc biotechnology also, you had some project. I believe, I don't know how much depth it was. And you have probably passed out in last 1 year and 1.5 years. So some portion of your college education is also under influence of Corona, like lockdown, projects might not have happened, right? You had projects in your BTech programs, I mean to say BSc programs, at least for some extent. Have you any project reports you have done? You must respond in yes or no.
No, sir.
Is it the same case? BSc program does not have any project?
No, sir.
I mean to say, not even some for few credits, small set, small segment period. No. If it is no, then this is really important that you should concentrate in MSc program. You will have one project. You have to submit your thesis. You have to do some sort of research work. I'm not saying the hardcore researcher. You cannot be. We are doing it for last 20 years also. We find it really difficult. But it's all continuous learning. And the learning starts from this semester itself for your MSc biotechnology program. And what subject we are going to study? You must be knowing that is research methodology and applied statistics. Truly speaking, if you ask me as a researcher, there is no methodology for research. There is truly no methodology for research because no rule you can fix for each problem, each individual, each kind of situations, how you are going to do in a lab, day-to-day basis, how you are feeling the lab is very important. And I am sure the theory will only guide you some framework, but until or unless you are coming into research, you cannot have. I will also share you some textbook. Of course, the question comes, sir, only learning is sufficient? No, we will also evaluate you. We will also have exam, right? And how much you know and appreciate how the research should be done. At the end of the day, you will also have exam systems. You can have no excuse for that. So that means I need to have some structured syllabi also, even if I don't like it for research methodology at least. But everything you will not get from the textbook. Please be assured. Everything you will not get from textbook. Textbook will give you some theoretical foundation, and I will definitely share those links in your Google Classroom. Okay, the classroom is created. I'll also share you the link for the classroom. where I'll be posting some study material. I'll be posting the lecture plans, how we should go, why we should go in these aspects. I'll be mostly taking this class. No pre-prepared PowerPoint will not be there. I will be writing on PPT. I will be describing on PPT, explaining until your classes is coming in campus. In campus also, we will have some PPT and PowerPoint presentation plus chalk and talk. All the methodology, including lab visit in our lab, is all welcome. Now, what you see the subject title, there is two component. One is research methodology and another is applied statistics. Now, if I say what is research, you know that many people, your seniors may be doing some research, your teacher might be doing some research. Everyone is doing research. Some research is fetching you Nobel Prize, yes, but all research are not fetching you Nobel Prize. It does not mean that all the research are useless, not like that. It is actually incremental updation of knowledge in a knowledge pool. You are doing some work, someone is doing some work, we are disseminating our knowledge, right? If you do some research yourself, and if you're not telling what you are doing to the community, then your research is no use, right? Some days you are very good researcher, you are doing marvelous things, but in confidence. And at one day, when some person who is doing the research, he or she expires, all the research is gone, because it is becoming very confidential things, no use for community, no use for science. So in research, communication is very, very important part. Whatever we do in our lab, we will have some protocol that I will discuss coming days, but we have to communicate to the community. And who are the community? Of course, we are not going to publish our research work in a newspaper, right? A daily newspaper, not definitely, because this newspaper is for all common audience. And research article is not for common. Yes, it is not for common people. It is for like-minded people who are inclined to the research, who are expert in the domain, or if it is a social research, to maybe to some extent, yes, for laymen also. But when the scientific rigor is too high, when we are dealing or detailing a complex theories, and I'm explaining to someone, we have to also communicate to the scientific community. We learn from others, right? How you are learning? You are reading some textbook. Who has written your textbook? Some of the author has written. We are learning from research papers. Textbook and research paper, research article, all those things will be integrated when we are talking about research methodology. So when I'm talking about any methodology, that means you will be expecting some protocol, right? How to do, how to deal the situations. If I ask you what is the methodology of cooking, you must be, if you are known, for example, I cook sometime, not sometime rather, I cook regularly for me. I know the recipe, right? For example, if I'm making some curry, so I will, maybe fish curry, I will clean the fish, I will chop the onion first, I will clean my utensils for cooking, I will put oil, maybe my recipe may be different from you. But basically there is of course a protocol, how I am following, right? I will not put the fish first, then I will put the oil and something, then I will switch it on the heating. No, this should be always systematic. Your research is also systematic. And we know the goal. What is my goal if I am cooking? Okay, I will have some fish curry, I will enjoy it. So similarly, when you are doing some research, you will also have a goal. The goal is, what could be your goal is all different. There is no unified goal. For example, one of my students is going to do a research, and she is interested to overproduction of biopolymer. Maybe biopolymer, you know, right? Some microorganism can produce polymeric substances inside their cell. They do when there is shortage of carbon source, nutrient is low, then microorganism actually build up these polymers inside their cell. Why they are building up? Any microorganism, any plant, if they are producing something for biotechnological purpose, they are producing for us? No. Plants is growing fruits. Is it for us? No. That is for their own purpose. Similarly, microorganism is producing this polymer because they will utilize this polymer after some time. When there will be no food, they will use like a storage. Now, what she is doing? they should have some objective. Any research will have some objective. If I say my student's objective, she wants to produce biopolymer more amount. How can you produce more? Then question comes. So objective is fixed. I should have some avenues. You may say, sir, I will look for some microorganism which can produce very high amount, efficient microorganism. Yes, I agree. Someone will say, no, sir, I will know I am very good in biochemistry. I know the metabolic pathway and I will change something and I will ask the same microorganism to produce more. Accepted. Someone will say, sir, microorganism is producing the polymer, but they are keeping inside cell. What if I make the microbial cell little porous so that they will release the polymer and it will be more easy for us? Yes. So you see, I'm having one big target, but I can have multiple avenues how I'm going to improve. Someone may going to improve the production of the microorganism. Someone may say, sir, the nutrient is too costly. Do you think I will use glucose every day? Glucose is costly. At least, can I use some waste material, wastewater or something for production of polymer? Yes. So actually, your goal is very clean. The domain is very clean. People works in many domains. You might be working for maybe extracting some plant metabolites, plant extract, and you want to test whether it is useful as a painkiller, or maybe you want to test whether this can be used for wound healing or burn, maybe some high degree of burn, can you treat it? So there can be many applications. This is one. For example, I'm talking about health related. I am actually environmental biotechnology, right? So most of the time, my example will come into some environment related things, but not necessarily. Research is going on every sphere. Someone is doing in our institute, you will find in our department, some people are working on animal biotechnology. They're modifying the animal cells, okay, animal cell lines, insects, they're trying to manipulate. Someone is going to produce some treatment for dengue virus. Rabies vaccination someone is going to do. So immunologists are there. All people, their goal is same. What is their goal? To improve human life, right? That is the only goal you can have for any research. Someone is developing mobile, someone is going for artificial intelligence. What is their goal? So that it will improve your daily life. Someday you have to go and going to managing something else. You can now do these things from your mobile. Transaction, you have to go to bank. Now you are coming it here in your fingertips. Any research, they also, do you think they does not require any research? Of course. If an LED light, do you think it is an innovation which has actually changed dramatically all those things? Power consumption has reduced drastically. So you see, we are planning for something and some other benefits can be there. Yes. So for example, if I'm saying the nuclear research, what was the original people when they started these models and identifying electron, neutron, proton, molecular level? What was their aim? To harness the energy. We have nuclear reactor also. We are having nuclear bombs also, right? So thing is that our objective is always to have something productive, either from plant biotechnology perspective, from animal biotechnology, or environmental biotechnology. As environmental biotechnology, if I do some research, I will try to do what? Maybe I will try to reduce emission. What I will try to do? I will try to remove the contaminants, contaminated soil. I want to clean them, possibly. We want to produce some energy, green energy, biogas production from waste. So your interest and your domain, I cannot fix. It is your choice, your inclination will be doing. But we will have some protocol. Protocol means just guideline. It is not a very strict protocol, which should be done in a research. So if I'm coming to a project, for example, you are coming to me or maybe someone guides and you are going to do some projects. Someone wants to do, okay, I want to evaluate the potential of the plant extract in anti-diabetic treatment. I want to evaluate. For example, if I give you this project that you want to evaluate all the localized plant species, maybe in your native, what are the different plants are available? I want to see, is there any plant extract which can be useful for diabetic? If this is your target, it is good purpose, right? You want to have some drug maybe. So if you have this problem is ready, then question comes, what you should do the study. So a big problem can have very small sub problem, right? Sub problem is what? First problem I will say, everyone will not actually, I am telling this way. Some other researcher you ask, some other scientists, they will speak different way also. There is no unified approach rather. So if you ask me this problem, what I will do or what you should do, first thing is that, okay, I will try to know what are the different plants we have in our region. So plant diversity in my place, I will try to find. Once I am having the list, what I will do, I will try to see, is there any traditional knowledge is there? People might be using some plants without knowing the science. Prehistorical peoples might be using. And those knowledge you cannot simply ignore or discard. No, it is non-scientific, not like that. They might not have known these things scientifically, but science is always there for every event, right? So if something people are using for long time period, maybe you might be knowing some traditional medicines are there. So traditional medicine might have some implication. There might be some compounds. That means you need to respect those aspects also. Biodiversity is there, traditional knowledge is there. So I will try to know which plants are already known for some medicinal values. I will shortlist them. Then question comes, then what we will do? I will try to make these plants, I will collect those plants, maybe plant different parts, leaves, or maybe root. Then what I will do? I will try to dry them up, right? Protocol wise, I will dry them, I will crush them, then I will put some water to extract it, right? So I can have different extract. Maybe I am trying to extract in water, I am trying to extract in methanol, I am trying to do. Then what I will do? So what is my objective? To test anti-diabetic property, right? Then what can you give? You can give to some diabetic patients also. Yes or no? Normally you will say, I will give to diabetic patient. But there is a problem, because science also needs some ethics should be always followed. You cannot say, I prepared some plant extract and I given to my neighbor. You cannot publish this kind of work, because science also, scientific reporting, scientific research also need a coherent and adhere protocols for privacy of the individual whom you are going to treat. Ethical committees are also there, and you cannot do any animal studies or human clinical trial. You cannot do. You need to be guided by regulatory framework. So if you know that research is being guided, if I'm doing with plant, if I'm doing with some animals, we're having animal house, but also we need from ethical clearance. You cannot kill the mice or rodent we are purchasing, right? Simply you cannot kill. You have to give count for what reason you are killing also. So every country, every institute will have its some regulation we have to follow. So research, when I'm talking about research methodology, these knowledge should be there. So we will be guided, we will be working something. So plant extract, if I'm finding, what I will do, because I do not have permission to give you, but I am having permissions for some animal studies. So is there any possibility in animal studies I will do? Yes, I will do animal studies also. But before going to animal, I will see in the, for example, in vitro, some studies can I do? Can I take some sugar or starch and I will put these plant extract and I will see whether the sugar level is reducing or not. So I will do some lab in test tube also first. So always our experiment will try to validate. First thing is on the test tube level, in the lab scale, without any animals, anything. Then we will go for microbial. Then I will test. Okay, for example, I find the drug is very much good in reducing blood sugar. Is it fine? In a lab, I am finding. Is it sufficient? No, it is not sufficient. We have to test whether this is safe. At least you test in first microorganism. You cannot go to human. You test on animal studies. You take some mice or guinea pig. If you are having more pronounced, you want to see more, you can say monkey. So closer to human being. So I can go from some studies, chimpanzees, these animals we can also include. Of course, you need guidelines, you need protocol for that. So basically, I need to know how I am going to study. Rather than what you are going to study, how I am going to study is more important. Now, okay, you have taken, for example, plant extract, you have studied in lab and you found it is good. But you are finding that no, when I'm injecting this to some mice, blood sugar level is not falling. What does it mean? Or maybe you may observe that the animals are actually being killed. So it may have something which can reduce blood sugar, but it may also have something along with it which can be poisonous or dangerous. So that means I need to have some protocol for analyzing its toxicity also, whether it is toxic. I need to have a protocol to measure what is the compound there, how they are working. Many questions can be done. And once we do the all job, we will try to find out the research and we will try to put it in a research paper, right? There are different kinds of research paper you have seen. I'm not sure. Have you seen any research paper? There are many publishers, scientific publisher. So once I will finish my research work, I will send a report of the research in a format. There is a formatting is there. I will send to a journal. Then journal will send my research work to another few scientists. They will evaluate whether my research is credible one or not, it is good one or not. Then they may accept my proposal and they will accept my articles. So my work will be accepted. If my work is having some flaws, many work can have many flaws, I will tell you. Your work may be rejected also. So basically, this will be guided by some frameworks, and I will try to make you understand how can you plan. So basically, I'll be using this course, research methodology, to make you how to plan. And many people say planning is fine, but many people actually fails in statistics. What is that? Let me tell you a very simple example. I find I am testing one drug in 4 mice. Okay, I told the blood sugar. I am having 4 mice. I am injecting in the 4 cases. I find 2 mice dead. In another 2, I find blood sugar reduced. Now you are in a dilemma, right? Whether it is good or bad. 2 mice has died. 2 mice blood sugar is reducing. So what you will claim? Is it a good or bad? So you have to take a decision, right? And your decision is not only your decision. You are only taking a decision from the scientific community. You are not you yourself. You are talking about science. That means everyone should accept your idea. So how to validate that? Out of 2, if 2 is dying and 2 is going, we can do this study by analysis. We will have some statistical analysis to validate whether can I accept these things or not. I'm doing some 100 rat and I find 95% of rat glucose level is reducing. Unfortunately, 5 has died. Is it acceptable or not? So these criteria, how can I compare? Or maybe you are having 2 plant extract from plant A and another is plant B. Then you are telling my plant extract is very good compared to the old one. That means you have to do a study, right? If I am telling that this plant extract is better than anyone else, what do you have to do? I have to prove it, right? I have to use the same plant extract. Side by side, I will also use the already known plant extract. I will see the result. I will compare them. Then I will make a comment. So basically, research methodology will tell you the protocol, and research methodology and applied statistics. The statistics is very, very important. Many times you people, I will tell you, many times, many researchers do very good work. Maybe scientifically work is very good, but they do not do statistical analysis of their result. And that makes their whole work less important, or sometimes the work is rejected also. So work is not only important, it should be statistically sound. Statistics, you know, right? Some extent you know statistics means. Statistics means any natural process can have some variability. We are going to see whether this variability is acceptable or not acceptable. If we say that this much variation is always there, if I say, what is your body weight? Can you say very specifically, sir, my body weight is 50 or 60 or 70 kilogram? Not very precisely. Today may be 71.512 gram. Tomorrow may be a little bit less. Yesterday was a little bit high. Then if I say, okay, I am scientist, I want to know very precise, then what you will do? Then you will say, sir, you see, I cannot tell you very specifically. But last 7 days, I have monitored my weight, and you see, this is my data. Then what I will report? I'll give you all 7 days data. No, I'll say for last 7 days, average weight of, for example, this student was this much kilogram, and the variation was this much, standard deviation was this much. That means I am more accurate rather than if I am telling you the average weight only. If you simply tell average weight, is there any importance I am getting? No. If I tell average weight plus deviation, standard deviation, that means I am giving you more information. I am not hiding anything. I am telling whole picture to you so that you can decide. So these aspects are very, very important, and in statistics we will be learning those things. So today you see, I have not taught you anything. What I taught you is that when I am going to do research, you need to have some protocol, but there is no strict protocol. Always everything is flexible because you are the researcher. You can decide, you know what you are going to do. You always have an objective. You have always a target, and you have to fulfill the target. For that, you have to do some experiments. So tomorrow my student is doing, for example, she is setting up some experiment now today. She is seeing this, that after 3 days, what she will do, she already knows now because she has planning that, okay, I'll put this microbial culture today, I'll put in Sekar incubator, I will incubate for 48 hours, then I will take out the flask, then I will centrifuge them. She has a protocol, but not very hard that 48 hours means 48 hour, 1 minute should not be allowed. Not like that. She is flexible. She can change. She can change the protocol little bit, but she has some vision how she is going to do. She is going to isolate this biopolymer. So how she is going to do? She has made a protocol. So research methodology is going to talk about how I am going to design, how I am going to plan the study. Not one case, maybe her whole thesis, how she is going to complete the whole work. So you should have a planning. That planning things we will be doing for the whole semester. For different example I'll be taking. And today, as I am telling everything very much, you see, there is nothing to note right for you today. But it will not be happening every day, because if I speak like this, like a political speak like that, right? Nobody is recording. Then at the end of the day, you will enjoy the class, but finally there is no take-home message, right? You should have something concrete out of that. So for that, I will also be writing some PPTs, whatever I'm discussing in the thought, and the recording is being made on every class. Not today, but before your next class, I will be also sharing you the recordings, right? 1 second, please. I will stop this class very soon. But before that, I also want to share my screen here. And I'm coming to your classroom. This is your classroom this one is your research methodology and applied statistics. For this batch, this is the class code. Okay, I think you can see. I will invite you all to join this class. I think I'm copying the link. I'll be pasting this link in your chat box now, and I will request all of you to join this classroom. Second thing, I will also share another important aspects. The whole class recording, let me tell you. Whatever class we are taking daily basis, the recording is being actually stored in Meet recording folder. I'm coming. Yes, this folder, all the classes recording will be coming. Okay, but what will happen? The whole folder is shared with everyone. Okay, you see, every day after I'm taking a class, one video will come and one attendance will come. I have shared the entire folder with all my students for all the batches. Okay, so I have no restriction on that. And this is already shared. I will get the link. You see, it is shared with anyone on the internet with this link can view. So I'll copy the link, but it will be little tricky. I'm also sending the folder link here. So all classes every day after class, the recording will be available to you. I will also post this link here also in your Google Classroom. Okay, so this is the class link always. So you will be landing in the folder itself. Okay, you will be coming here. Only problem is that you have to identify, because many other class recording is also there. You can identify by title and you can check your classes regularly. I'm stopping the presentation now, and even I will stop the class. I have shared the link. Please join in this group. Oh, very good. All 8 people have already used the link and joined. Nice. So all of you please join here, and the recording link, you please remember every day you can have the recording here. And most important things, don't be silent in the class. It is not that I'll speak for 45 minutes and you will listen like a 45-minute some web series, right? Not like that. You can always speak. Any kind of doubt regarding research methodology, you can put some example. Sir, I want to be, for example, if I'm going to do this study, what could be the possible planning research methodology? Don't take this class as theory class alone. You try to understand that when I'm going to do some research in coming semester, I mean to say final semester, I have to face many problems. I have to be very good in scheduling. If you cannot schedule what is going to happen after 2 days, 3 days, you will always fail. You have to foresee some 2-3 days, at least 2-3 days at your level. We need to see at least for month level what I am going to do, or year level what my student is going to do after 1 year. I have to plan right now. Accordingly, she has to move stage wise. So research methodology will be talking about that. Some class material and books also I will be sharing. You go through the books, but you need not be very serious about the books, right? I'll be talking in this class, I'll be putting some recording, and I'll also be sharing some PPT, PowerPoint presentations file. You can take it, and research is all mostly on internets, on journals, and books will be there. You need to know what are the different publications where we are publishing, where your teacher is publishing, where we are working, how to communicate, how to do the work in lab. So it is actually practical, and we have a theory for a practical, right? So you should be careful on this. So I wish all the best for this semester.
Thank you, sir.
Thank you.
Thank you, sir.
Thank you.
Thank you, sir.
Thank you,
Thank you,
sir.
sir.
Have a good day.
Thank you, sir.